So my plan today is to talk to you more about plant microbe interactions, and then I can leave a little bit of time to do any last minute questions or clarification on exam content. I have a pretty solid draft, so I'll talk to you a little bit more about that in a little bit. So we've been looking at this concept. So the roots, when you dig them up, if they have these tumor-looking things, that's not a bad thing. That's actually the root nodules, and that's really the plant made that. It's like a little home for the microbes, all right? So when they start entering into this relationship, there's chemical communication going back and forth, and the plant is stimulated to go ahead and start shifting its morphology to create these little nodules. So if you were to slice that open, it's lots and lots of plant tissue, but then there's lots of microbes in there too, these bacteria. And so the idea here is that the bacteria, they get a nice home, they're sheltered, and then they're capable of fixing nitrogen for the plant and giving some of that usable nitrogen back to the plant. So the usable nitrogen is the idea that the ammonia that gets created will get used by the bacteria, but it also, a portion gets given to the plant, and they're able to immediately start working with that and take advantage of it. So we've been looking at this idea, and I think we were talking last time about the idea that there has to be this chemical communication. They have to find each other. I mean, the soil environment is pretty vast and lots of different microbes there. The plant has to find the right one. It's a pretty specific interaction. So, for example, a pea plant will only form this relationship with certain types of microbes, very limited. So it's not like any old nitrogen fixing bacteria can hook up with a plant. It's pretty specific. And so there's different chemicals that will be excreted by these different plants to attract their particular partner. And then the bacteria have to be able to sense that. So this is that so-called positive chemotaxis. That's just the idea that the microbe is picking up on the chemical signal. They start to go up the concentration gradient, which will lead them to the plant roots. And it depends on the plant. It could be a homoserine type chemical. It could be a pigment like flavonoids or isoflavonoids. So there's different plant chemical signals. In general, we say that the bacteria are secreting nodulation factors, which can vary, but they're a certain class of chemicals. We looked at them last time. And again, this is just to get you to think about the idea that this can shift, this chemical structure can shift. You can have repeating units to give slightly different nod factors. And then when the bacteria finally get to the roots, then they're like, yeah, here I am. I'm a friend. I'm not a foe. And let's enter into this relationship. And then the plant will start to respond to that by usually curling their roots. Their roots will start to branch a little bit. And I think this is where we left off last time, with the idea that the root hair here is curling and kind of hugging and scooping up the bacteria so that they can start entering into a partnership. So now the goal is the bacteria need to gain entry and get into the inner part of the root tissue. Right now, they're just interacting with root hairs, which are just modified epidermal cells. So now they have to get down that root hair and get into the root tissue, deeper into the root tissue. And when they do that, they have to do it carefully so that the plant doesn't get triggered to try to defend itself. And so what happens is the bacteria will start gently releasing enzymes to like partially degrade a section of the cell wall, because plants have cell walls. So they're going to be needing to get past that. So they have to degrade that a little bit. And then what they do is they won't attack or try to break down the cell membrane, because that would trigger the plant to start defending itself. So basically what they do is they enter into the root hair, usually near the tip, and get past the cell wall, and then they bump into the cell membrane, so to speak, and they just start pushing that ahead of themselves as they migrate down the root hair. And so the cell membrane, as it's getting pushed ahead, it kind of invaginates around the bacteria, and they start to call that process the infection thread. All right, because when you look at, I'll show you some pictures, you look at it under the microscope and it's just these bacteria kind of in single file, one right after the other. And the lead bacteria is pushing that cell membrane ahead of itself. And in that manner, then the bacteria are never breaching the cell membrane. And so the plant is like, okay, yep, you're being polite, you're respecting my boundaries, and I'll let you keep coming in and we'll set up this partnership. And so they actually call it, unfortunately, the invasion process, which is kind of a negative connotation, but that term stuck. So they call it the invasion portion of this interaction. And then when you think about the bacteria going down the root hair, they call that the infection thread. And so here they're trying to show that a little bit more, where the bacteria are encased within the cell membrane, so to speak, all right, as they migrate down that root hair. So they're always bounded by a plant-derived membrane. And you see that once they start making it into more of the parenchyma cells of the plant, deeper, beyond the epidermal layer, they start to have these fusions of the membrane with the cell membranes of those cells. And so no matter what, as the bacteria are migrating into these cells, they're always surrounded by membrane and never breach that. So the lower images are showing some nice images of the infection thread process. And you can see how there's lots of bacteria that are making it deeper into the tissue of the root. Here's another really nice image showing the bacteria moving down the root hair. So this is a nice strategy. The root hair is, it's pretty dainty. It's relatively straightforward for the bacteria to, get into there and start migrating deeper to the inner parenchyma cells of the root tissue. Okay, so once they get into the root tissue proper, into the parenchyma cells, then they have to set up camp and establish a home. As that's happening, that's the area that starts to morph into that nodule. And so that portion of the parenchyma cells of the root will start to morph and get that growth of the nodule. And so this step we call the release, because they're releasing into those parenchyma cells of the root and the nodules. But again, they're always surrounded by the plant membrane. And then what they see is a pretty dramatic shift in the bacteria. Once they get to their final destination in the root tissue, they start to enlarge dramatically. And when they do that, then they start referring to them as bacteroids. So if you hear someone say, oh yeah, look at those bacteroids, they're looking under the microscope into the nodule and seeing these really big plump bacteria. So they've greatly enlarged. And you might see in the literature that they'll talk about the bacteria are surrounded by a peribacteroid membrane. That's just this plant-derived membrane that's always surrounding the bacteria. More recent publications are starting to call it the symbiosome. That membrane, they call it the symbiosome. So as the bacteria are enlarging, you still see that they're encased in that plant-derived membrane, and then they start referring to it as a symbiosome enclosure. And then the other thing that has to happen is into those nodule areas where all the bacteria are, you have to have good vascular connections. So the plant has to get its vascular tissue embedded into the nodule nicely so that the bacteria can get fed. So the sugars will come via the phloem of the vascular tissue to the bacteria. The bacteria will get that ammonia through the vascular tissue to the plant. And so it's like an internal highway system. So the nutrient flow can go back and forth between the plant and the bacteria. So if everything goes okay, you'll start to see the nodules forming on the roots. The lower picture on the right is just showing all the bacteria hanging out in a nodule. And then the drawings up top are trying to show the migration of the bacteria into the different parenchyma cells that are starting to become the nodule area. And they're trying to show that they're always encased in membrane. Here's a nicer image, more detailed with electron microscopy. And so these are the dark gray, that's the bacteroid. So you can see they're quite big and plump. And then the membrane surrounding them is the peribacteroid or the symbiosome membrane. And so you have the bacteroids have really shifted in their morphology and then the membrane surrounding. So very dramatic. But it's all good. It's a good thing because you're going to get that nitrogen fixation cooking along once they get established in the nodule. The downside of that is that it's expensive. It takes 16 ATP just to fix one dinitrogen. And so I would like you to know the general idea here. So in order to take dinitrogen and break it down to get ammonia, you do have to expend 16 ATP. That's a lot. And so when plant physiologists are measuring the cost of this to the plant, they often refer to it as how much carbon does it take to do this? And so like with soybeans, they've done lots of studies on soybeans because they form this relationship with bacteria. And they say 12 grams of carbon is required to fix one dinitrogen, 1 gram of dinitrogen. That's a fair bit of effort by the plant, but the benefit outweighs the cost because this relationship has been going on for millions of years and it does benefit the plant greatly. So, there's many of you are botany students in this class. You know that plants definitely have a need for nitrogen. There's a high demand for that. And so if you can form this if you're a plant and you can form this relationship, this is pretty awesome for you because you're getting this ready-made nitrogen right there in your tissue. And it is you're far less likely to have stress of a nitrogen deficiency. When you think about soil systems, soils can vary from, week to week in terms of how much nitrogen they have available. And it really does depend on, especially if you're if you're a plant stuck in an agricultural field, you're at the mercy of someone running that field and applying the nitrogen. And sometimes that doesn't always go so well. So having this relationship is a great thing. Scientists are really excited about this idea because if they can better understand this relationship, and they have a pretty good handle on it, but they're thinking, could we create plants that don't normally do this with bacteria? Could we get more plants to do this, more plants to partner up with bacteria? And so that's something that they're looking at. And then there's other strategies too, I'll talk about a little bit later on just how they're using free living microbes to also fix nitrogen in soil systems and kind of boost that all in the soil. But basically, if you could get more plants, especially agricultural plants, to do this with their associated, bacteria, the amount of nitrogen fertilizer you'd have to apply to the system, the soil system, would greatly diminish. So that would be a huge cost savings. Plus, it's more sustainable. Getting nitrogen fertilizer made is not great for the environment, and it's a lot of energy costs to that process. So scientists are really excited about this and have been studying this for quite a while now, trying to figure it all out and see what they can do. So there's one thing about the enzyme that is involved in getting that dinitrogenase enzyme. When you look at it, it's a very funky enzyme. It's the one that's breaking that triple bond. It's unfortunately really quite sensitive to the presence of oxygen. It tends to fall apart. It's a multi-subunit protein, and when it gets exposed to oxygen for too long, it just kind of falls apart and stops working. And of course, that's kind of disastrous. You don't want that happening. And so, and then you think about some of the bacteria. When we looked at some of the bacteria that can fix nitrogen, if you look here, there's aerobic bacteria that fix nitrogen. So they have to have oxygen. And yet their dinitrogenase enzyme is sensitive to oxygen. So this is a real conundrum. How do you deal with that? And so if you have to breathe, so to speak, but the enzyme you're using can't be around the oxygen to any significant amount or time, then you got to come up with some kind of strategy. And with this, with the bacteria that hang out with plants, there's a nice little strategy that has evolved. And it's this protein, this oxygen binding heme protein. It's called leg hemoglobin. And that's what's giving rise to that pink coloration to the nodules. If you dig up a plant and see that some of the nodules look pink, you know that leg hemoglobin is there in abundance and it's doing its job. It's very similar to human hemoglobin, which carries oxygen to our cells and feeds our cells oxygen. So they called it leg, named after legume plants, so leg hemoglobin. So what this does is the plant, they thought originally that the plant and the bacteria partner up in making leg hemoglobin. They thought it was a partnership process, but more recent studies have shown that the plant seems to make all of it for the bacteria. And so anyways, the plant is synthesizing this, and what happens is it allows for oxygen to be bound to the leg hemoglobin. And so if it's bound, it's not going to interact with the enzyme, but it can release the oxygen. The leg hemoglobin can slowly release the oxygen to feed the bacteria oxygen to keep the bacteria alive, but it does it in a very controlled way so that you don't get a buildup of oxygen around the nitrogenase enzyme. Does that make sense? Okay, so you don't have this big flush of oxygen, free oxygen just floating around this nitrogenase enzyme. The leg hemoglobin has control of it. It says, okay, I have it. It's part of my, I've attached it to myself, and I'm just going to slowly release it so the aerobic bacteria can still get oxygen to stay alive. Your enzyme will stay safe from oxygen. So that's a really nifty adaptation and really solves the problem with the bacteria that hang out with plants. The plants are nice enough, so to speak, to make this leg hemoglobin and have that available. Free living microbes, though, that are aerobic. Whoops. Not that one. Not that one. Where'd it go? Free living. Let's go back here. Free living. So free living, aerobic nitrogen fixation going on. They have to have other strategies. And so often what we find, especially they've studied azospirillum a fair bit. What they find is that azospirillum is super efficient at using up oxygen quickly. And so it has a very high respiratory rate in the sense that it is using available oxygen very quickly and running an electron transport chain process. And when you do that, you convert oxygen to water. And so if they have a high respiration rate, they're constantly making sure oxygen is involved in respiration and not just kind of free floating out there, where the nitrogenase enzyme is in the cytoplasm. So that's one strategy that they can use. Anabina and nostoc, on the other hand, when they're free living, they're also creating oxygen because they're doing photosynthesis. So that's a big problem. And Anabina, they've studied that one quite a bit. That one, instead of being really high respiratory rate, they actually sequester their nitrogenase enzyme. They tuck it away in a special cell. And when you look at Anabina, they look like a pearl necklace. And so they have one of those pearls, so to speak, is a specialized cell where they do nitrogen fixation. You look in there, there's no photosynthesis going on, no oxygen there. And so they spatially sequester where they do nitrogen fixation, and then all the rest of the cells in the chain are busy doing photosynthesis and all of that. And so there's different strategies depending on the microbe, how they go about protecting their nitrogenase enzyme from oxygen. Any questions on the biological nitrogen fixation idea? I know that we also looked at this in lab, but this is a little bit more of the process. Oh, by the way, has anyone, if you've ever eaten a Impossible Whopper, Burger King sells the Impossible Whopper, and it's by the Impossible Company. I went to a plant biology conference right before COVID broke and went to a symposium session where they were looking at I don't know what the whole session was about, but anyways, the scientists that came up with the Impossible Whopper. So it's all vegetarian, but it mimics meat like a hamburger. And so it was really interesting because they were talking about how they were so successful. If you've ever, I have eaten one and it does taste like meat. It's amazing. And so they were going through the whole process of how they came about doing that so successfully. And it was this idea of using like hemoglobin. Because when they were doing, they would get people together and say, taste this, what do you think? What do you think? And they'd rate it and, do their feedback. And then they would take that and like, no, this doesn't taste like meat. And what they realized is that our human physiology and taste sensibilities, we pick up the reason we say, oh yeah, this is meat. One of the big reasons we say that when we bite into meat is we taste, although you don't like consciously probably think about this, but you taste blood. And that's why kind of our primeval, like, yeah, that's meat. We've evolved to eat meat and way back. And so here, this idea of, okay, so like hemoglobin hemoglobin is in blood of animals that you eat if you choose to eat animals. And so the leg hemoglobin is going to taste very similar. And so that's what they started putting into their recipe. And that made all the difference. I thought that was genius. I give them a lot of credit for that because that was so, and then they can still say it's all vegetarian. They didn't put in heme from animals. They put in leg hemoglobin, which is again vegetarian. So a really interesting story. It was nice that they were talking about that whole process from start to finish. So, although I haven't looked at the totaling, well, I have looked at the ingredient list, but I think in general, nutritionists say if you're going to eat Impossible Whoppers or some of the meat substitute things, they're not really like something you should do on a super regular basis in terms of health. It's more of a treat, I think, is how they are saying you should, if you're going to eat that, don't do it frequently because it's some of the ingredients, maybe the fat content, I don't know, but just keep that in mind. So just because something is vegetarian doesn't mean it's absolutely healthy for you. Okay, anyways. All right, so another topic I'm going to introduce, and then we'll switch into talking about the exam in a little bit here. But I wanted to at least introduce this concept. We've kind of been talking about it because certainly nitrogen fixing and mycorrhizal fungi, they all fit into this category. But it is something that you see a lot in the literature, and depending on what year it was published, newer publications are using the acronym PGPMs, plant growth promoting microbes, but you'll often see other like PGPR for rhizobia. So those are so again, this is when they say that, it's just saying they hang out with roots. Okay, rhizo means roots. That's all they mean by that. And then you often see PGPB, and the B is for bacteria. But So those are earlier publications. They're using PGPM now because they're recognizing fungi are involved in this. And really, PGPM is more of a bigger umbrella term because bacteria, fungi, and then rhizobia is specific to the roots, but we know that there's plenty of PGPMs that hang out on leaf tissue as well. So it's not just for those that hang out with roots. So the PGPM, I think, is actually a better term. So there's been a lot of research in PGPMs and looking at how can we use microbes that coexist with their plant host or just hang out with plants and use that ecological knowledge to our advantage, especially for growing crops. And so in the agricultural realm, this is catching on. Now, not that long ago, this was a fringe science. When I started working with this concept back in grad school, when I'd go to conferences, I'd have senior scientists at the conference kind of um, be dismissive, I guess would be the way I would describe it. They would be a little bit dismissive because they were so confident that we had all these lovely pesticides and chemicals that we can use and just spray the heck out of the fields and that will support the plant. And you don't need to worry about all these microbes and what they do in terms of the non-pathogenic ones. You know, they were very much zeroed in on the pathogens. And we have all these chemicals for getting rid of pathogens, so we don't need to worry about it. And so when I was first involved in this, it was a little frustrating, but as the years have gone on, and especially now, I feel validated because there are all kinds of scientists around the world now that are very excited about this idea and that they recognize we can't keep using all those nasty chemicals. And in fact, there's been bans on some of them, right? Some of them have been outlawed. And so then the plant biologists, especially the people who are growing crops for a living, started to get really worried because if you don't have those chemicals to kill off your pathogens, now what do we do? And so the other option would be one option. There's several ways to attack this issue of keeping your plants healthy as you're growing them, but one of those methods is this idea. Can we use the ecology of this interaction of microbes hanging out with the plant to support the plant? They're looking at not just fighting pathogens or helping the plant fight pathogens, they're also looking at kind of like preventative medicine. You know, you're always being told, drink lots of water, eat well, keep your immune system strong. And we know that microbes do help keep the plant's immune system up and running and efficient. And so having those partner microbes is important for that too. So a lot of the new science coming out, the results that we're seeing from nice studies are really starting to open up this world of, wow, these microbes are doing a lot for the plant. And can we take advantage of that and better help support the plant? And so, for example, now with synthetic biology and genetic manipulation of microbes, some of these companies that are starting to come about are looking at, okay, can we understand this? Is there a way we can tweak it to better get it, a little bit more efficient, more for what we want, specifically that organism, that microbe to do. And so this was a publication not that long ago, I think about 5 years ago. Pivot Bio is the first company to offer U.S. corn farmers a new probiotic for plants. And they're actually starting to use those kinds of terms. They talk about probiotics for the use of plants. So it's an interesting idea. And so startups, the idea here that there's all these new companies coming out that are looking for, I don't know, Bayer aspirin. So this is a huge company, biotech company. They certainly make Bayer aspirin, but they make all kinds of other things. And so this idea of self-fertilizing crops with the help of designer bacteria. So what they're getting at here is they're zeroing in on, can we find bacteria that we can, in this case, put into our fields that we grow corn in and get them to fix nitrogen. even if they're free living. Okay, so corn does not form a relationship in its roots with bacteria that fix nitrogen. Doesn't have that capability. Corn, as you know, is a huge crop in the United States, more than it should be, but that's just how it is right now. And so corn is what they call a nitrogen hog. It needs a lot of nitrogen through its growth season in order to form lovely corn on the cob. And so those fields are intensively managed with lots of nitrogen fertilizer. And that's expensive and it's not terribly sustainable. And if you do it incorrectly, it can create a lot of damage to the soil system, including killing off a lot of microbes. Because it sounds strange, but for inorganic fertilizers, if applied incorrectly, can actually do a lot of harm to the existing microbes in the soil, to the point where they can refer to some of the soils that have been so damaged. They call them microbial deserts. There's not enough microbes in the soil to really support the plant. And so it's a big problem, and we're starting to recognize that more and more. And so now they're like, okay, well, if we can understand the microbial ecology, can we, work with them, manipulate them somehow to get what we want to have happen? And in this case, is to get the nitrogen fed in a more sustainable way to the plant, to the corn plant. Okay, I think I'm going to stop there because the next several slides is going to take a little bit of explaining, and I wanted to leave some time for you to think about the exam, and we can talk about that a little bit. I'm going to stop share there. All right. So on Monday, when we were going over exam material, I think we were going through slide sets, and I think we made it to the epiphyte endophyte one, but then we had to shift over to lab. So let me pull that one back up. And let me do this. Okay. So this exam is tomorrow in class, and there's no take-home portion. It's working out, though, that there are more points coming from the essay than the multiple choice. And so that's primarily because the multiple choice isn't very long. Currently, there's 6 essay questions. What I would urge you to do when you get into the essay questions, some are worth like 4 points, and then some are worth more. So use that as a judgment. If it's 4 points, that usually means a handful of sentences will get you there. If it's 10 or more, that usually means it needs a little bit more explanation. And, the questions, when I write it, that I have more than one sentence as a question. It'll be, and I've tried to break it down. I'll say like, first answer this question, then answer this question as part of one essay question. So the other thing I want to caution you on is that take it very methodical and just answer one aspect of the essay question at a time. You can even number it out or letter it out and just bullet point it out. I don't need fancy paragraphs. I don't need that. Okay. Do what you need to do to make sure that as you're answering the questions, you can say, yep, I've answered every part of this question that was being asked. Okay. So that's a big caution, because I've seen students not do that and just write this big long paragraph, and then they can't easily tell if they've answered the whole thing. So I encourage you to actually, you can do bullets or whatever. I don't need fancy paragraphs. And then as you're taking the exam and you're doing the essay portion, if you are like, is this enough? call me over and I can scan it and let you know. I won't, I'll guide you on what I'm looking for, how much more I might need. Okay. So it's doable within, I set it up for an hour. Hopefully I'm going to get here right at 10 to. Hopefully we can start right at 5 to the hour and go to 5 to the next hour before the next class comes in. So I've designed it that way. So try to get here on a little early and then you can start early. Um, so also I wanted to also say that some of the essay, you have options. Like I'll say, choose this, choose one of these, and I'll give you like options. And so you do have that little bit of a luxury where, you don't have to necessarily answer everything in that question. So again, read the question carefully, because if I say choose one or pick one, that's literally what I mean. I don't want you to answer everything. Okay. So pay attention to that as well. Okay. So questions on what I was just talking about with strategy and format? Okay. Yes.
What kinds of questions are you planning on asking for like the essay, like just kind of like biological processes, like nitrogen fixation stuff?
Well, that nitrogen fixation won't be on the exam, but like this slide said, epiphyte, endophyte, right? So, for those who've had classes with me before and have done essay questions before, you might remember that I'm kind of a fan of compare and contrast when I ask you essay questions. So this would be a good compare contrast, right? Epiphyte versus endophytes, very different parts of the plant body, very different life styles. So thinking about, being more thoughtful about, well, okay, what are epiphytes? Where do they live? If I was an epiphyte, what would I be faced with? How would I survive? Same with endophytes, so looking at their interactions with the plant, depending on where they're located, so their ecology. So those types of ideas. Another compare contrast, when we talked about mycorrhizal fungi, we went through several different types of mycorrhizal fungi. So those, what's similar about them? What's different about them? Who do they hang out with? Is there anything unique about how they hang out with their plants or how the plant interacts with them? So those types of ideas. Um, so I would encourage you today as you're studying and to type things out. That's what you're going to be doing tomorrow. Type things out, like see what you can do in terms of some of the essay questions you think I'll be asking. And when you're studying for essay questions, of course, you need to do a couple of things. You need to be able to organize your thoughts. And I don't know how many times I got bit by when I was a student. Oh, yeah, I know that, but I never typed it out or wrote it out. And then when you come in on exam day, it takes you more time because you didn't practice it. It's sounds maybe like a picky detail, but it can make a world of difference. So back when I was a student, we didn't have laptops. So it's all hard copy paper tests. So I would write everything out more than once before I walked in on exam day. Luckily, typing, most of us can type fairly quickly, so that helps. So there's that. And then, um, where was I going? There's another thing. Essay questions. So practicing, typing it out, organizing your thoughts. There's another thing I was going to say. It just wafted away from me. Oh, just being succinct, learning how to be succinct. Okay. More is not always better. Like I said, some of these questions you can answer in one sentence. The one, if I say, A, tell me this, one sentence will get you there. B, tell me this, maybe one or two sentences. So being able to practice that before you walk in will save you time for sure. Any questions on the epiphyte endophyte slide set?
You know, like that names already, like.
No, no. Not terribly big on names. It's more the concepts. What are they doing? What's significant about how they live their life? Okay, well then I think the last slide set that's on the exam is the slide set we're covering currently on we talked about what is the rhizosphere, and then we started getting into how microbes in the soil system, especially right near the roots, are so beneficial for the plant to be able to get access to, especially to minerals. They really do need microbes in the soil for that. That's why if you kill off microbes, if you use all those chemicals that they're sensitive to, you're going to have a plant that could have all these lovely minerals in the soil, but they can't get access to them very readily without the help of microbes. And then we started looking at mycorrhizal fungi. So again, thinking about, we covered different types. We had the ectomycorrhizal fungi, the endomycorrhizal or the VAM fungi. And then we talked about the fungi that hang out with orchids and that concept of the mycoheterotrophs, where the orchids are kind of taking advantage of the fungus, at least for a while. So again, thinking about how these microbes are interacting with their plant host. What do they do? What do they look like when they're hanging out with the plant? There's some similarities and differences with these different types of mycorrhizal fungi. I did say that the terminology is important. So when you're looking at terminology, so for example, I asked you to get familiar and comfortable with the terminology, the mantle, the vesicles, the arbuscles, of course, the hyphae coming off the root, Hartig net. All right. So that's all fair game. Go away. Any questions? What about questions on any of the other slide sets? Yes. Ending. We are ending. I think it's slide 42, correct? Where we're not going to do nitrogen fixation on this exam. Yes.
We're going to go over the video, like 1:30 slide. We're supposed to know about that.
The slide on 42, you mean? Oh, it supports what I talked about when I was talking to you the other day about concepts on slides 38, 39, 40, 41. Those types of concepts are similar to what she was covering. So I won't explicitly ask a question from that video. I would have let you known if I would have told you. But that was more for, it's a very nice talk. She's a very elegant, eloquent speaker. And makes her points very nicely. Yes,
Take home portion.
no, no take home. It's a smaller, and it's not even 100 points, I don't think. Right now, it's not even 100 points. It's somewhere between 90 and 100, I think. Yes,
Ask a question about different types of degradations.
no. Mm-mm. No, it's more of the main point I was trying to get across there is microbes rule when it comes to degradation of complex polymers or polysaccharides. And very often fungi are greatly important, especially with some of the like lignin and cellulose, and they're hugely important. But I didn't go, when we were as a group going over it, we didn't really get into nitty gritty of that. I was just trying to emphasize how when you look at the decay portion of the food web, you really have microbes. If it weren't for them, our forests would be piled high with logs just in the forest that never got broken down. So super important. Other questions? All right, so I will be looking at my email into the evening tonight. So if something pops up and you're like, you want clarification, send me an email and I'll try to get back to you as soon as I can. Same with tomorrow morning before 10. And I'll try to get here right at 10 to 10. So that as soon as the previous class gets out, we can get in. Okay, so as you come in, please be quiet. You normally are anyways, but just be aware there's students taking tests probably if you come in a little late. Okay, that's all I got.